So good afternoon, ladies and gentlemen. Thank you very much, organizers, for inviting me and giving me an opportunity to talk about pre-implantation genetic testing of monogenic diseases, which is important for all couples at the risk of a rare disease. I would like to disclose at the beginning that I am an employee of GNT Labs, which is a laboratory specializing in the provision of genetic testing services for fertility clinics. And to start a talk on PGT, I think it's important to recapitulate the important numbers on rare diseases you heard in previous lectures. I think it's important to understand that nearly 3/4 of them are of genetic origin, which makes a sum of roughly 5,000 diseases of genetic origin. And it's important to understand that the knowledge of causative gene and mutation is an essential prerequisite for both prenatal and pre-implantation genetic testing. For how do the couples at the risk of rare disease knows about this risk? So the first possibility is that they have a live birth of an affected child or fetus with the rare disease. Second option is that they had a family history of rare disease and they know this from the relative through the genetic testing. And finally, the final option is a preventive genetic testing, like preconception carrier testing or expanded carrier testing, what we heard in the previous lecture. So they already know the information about this. This, in practice, in daily practice, in our lab, is mostly the case of infertile patients coming to fertility clinics because they have an access for genetic testing prior to IVF, and often a carrier test is performed at them, and they are informed. 1 or 2% of them are at the risk of conceiving a child suffering rare disease. So what are the options how to avoid genetic disease in children? So the first option for the couple at the risk is to simply resign on having biological children and adopt a child. Second option is gamete donation, and there is of course an option of prenatal testing and possible pregnancy termination, which has to be accepted by the couple prior prenatal testing. Otherwise, the prenatal testing is basically useless and has no sense. And a final option is undergo assisted reproduction, IVF, followed by pre-implantation genetic testing. So speaking about pre-implantation genetic testing, we should understand the current state of this method. So the PGT testing is the genetic testing of the early embryo, which is performed prior transferring this embryo to the uterus, so prior to transfer. And mostly today is performed at the blastocyst stage, when the 5 to 10 cells of trophectoderm are taken from the embryo. Embryo is subsequently vitrified. Then we have time for genetic testing of this sample, typically up to 1 month. And then after the genetic testing is finished, then the frozen embryo is transferred in the following months. So briefly, something about the history. PGT for monogenic diseases has started in 1989. It was by Professor Handyside and colleagues who did the first PGT for monogenic diseases. In pioneer times, it was about blastomere biopsy, so it was the challenge to make a genetic diagnosis from the single cell. They tried to keep it as simple as possible, so the first PGT was about simple sex determination or detection of a specific mutation, which was of course very dangerous because there is always a risk of allelic dropout, so you are always at risk that you are missing one of the 2 alleles. And in fact, out of the first 7 diagnoses, there was already 1 misdiagnosis in the cases. So after this pioneering stage, the second stage started in 1996, where PGT haplotyping was introduced. So basically the direct diagnosis of the mutation was supplemented by the linkage analysis using STR markers, and this eliminates the risk of allelic dropout, make the PGT diagnosis accurate, or nearly 100% accurate. It was possible also to link PGT for multiple factor, for example, even with the HLA typing, and techniques of whole genome amplification were introduced, which enabled later also to analyze aneuploidies along with the genotype of the embryo. So, very important thing that there was in 2012 there was a transition from cleavage stage biopsy and fresh embryo transfer to blastocyst biopsy, freeze-all techniques, and frozen embryo transfer. And after 2014, when karyomapping was introduced, usually PGT is coupled and combined with testing for aneuploidies as embryos, because this is important to increase chances of the couple for the healthy pregnancy and increase the success rate of the treatment. So actually, Professor Handyside was an important person in always when the PGT transitioned from one era to another, and we are quite happy to collaborate with him. And karyomapping is the current tool which is used. It's universal. The most important thing that patient and gene-specific setup could be avoided, and it gives you an opportunity to do simultaneous PGT-A, SR, and couple all these testing together into one test. So I think I should introduce you a little bit more karyomapping as a technology. The technology is based on SNP array. We already heard about arrays in previous talks. The SNP arrays today are currently used for cytogenetic testing to detect unbalanced microdeletion and microduplication, which also represents a significant part of rare diseases, actually. So the technology is actually coming from here, but could be used also for genotyping of the embryos. In 2022, we changed human karyomap bead chip for global screening array version 3, which is cost-effective and cheaper, and basically couple available products from different companies like MD whole genome amplification from Quagene, then chips from Illumina, scanner from Illumina, with the software developed together with Professor Handyside, which enable us very comprehensive and detailed aneuploidy detection on top of genotyping or karyomapping. And we are quite happy that we put together a manuscript, and the manuscript is already available as a preprint to be downloaded. So if you want to enjoy it, I encourage you to read it, because you can learn more about this fascinating technology. So how karyomapping works? So karyomapping is based on indirect linkage haplotype analysis. So instead of detecting a specific mutation, we try to specific alleles which are on the same strand of the DNA, and this is called linkage analysis. The important thing is that it's accurate, so it eliminates the misdiagnosis risk caused by ADO, which would be the case of the single allele detection. But finally, you can basically perform this linkage analysis for any position in the human genome, which means you can do this PGT haplotyping by karyomapping for any gene of interest for every mutation you have. And because you have a genome-wide SNP data, you can easily do aneuploidy detection. So there is a diagram briefly showing the principle of the karyomapping. You have affected child, typically, which you can use as a reference, and the paternal haplotype, which is shared by father, when it's mutated, it's the blue haplotype. The other one, which was not passed to the reference, is the red one. You can perform the same for the mother. So the haplotype shared between the reference child and mother is the yellow one, and the one which was not passed to the embryo without mutation is the green one. And then you, after establish this phasing, this haplotyping analysis, family analysis, you basically compare the embryos with the reference, and the embryos which has the same haplotypes as the reference are those which are affected, homozygous with the mutation, those which having or inherited completely different haplotypes from parents are healthy embryos, and then you have both combination for carriers which are healthy in case of the autosomal recessive inheritance. So there are cases where typically we need a reference sample to perform karyomapping, which is usually child or prenatal sample, like chorionic villus sample amniocentesis, or grandmother, grandfather, or aunt, uncle from the family. But also in cases when no reference sample is available, we have an option to perform direct mutation detection using amplified DNA from embryo. And we can use Sanger sequencing, qPCR, long-read sequencing, and all other methods. And then basically we establish the phase by this direct detection of the mutation in the embryo, and then go back and support the diagnosis data by the indirect linkage analysis. So that's the universal principle of karyomapping. So this is the nice dashboard which we have today. We have here for embryo samples. We have available QC metrics from the SNP array, brief overview of the aneuploidy testing and sex of the embryo. We have information on origin of aneuploidies, whether they are meiotic or mitotic. So mitotic are those which are basically mosaic then in embryos, and they are linked with the possibility of healthy live birth, and this embryo shouldn't be discarded from the transfer. And of course we have the result of the linkage analysis. So this is the color of the haplotypes, like I introduced you on the previous slide. So we have basically 3 dots there at the end, which means markers proximal to the gene, in the gene, and distal from the gene, which are basically then telling you whether the embryo is sharing the reference haplotype from the mother, or reference haplotype from the father, or the opposite haplotype, so non-reference haplotype from mother or father. So this is the detailed diagram on this, and we can use it when there are crossing overs. This is not that important. This is the detail on aneuploidy testing. And this is doubled, sorry. And finally, to some numbers. So in the last 9 years, we performed a PGT for nearly 1,000 unique patients. Typical patient has the autosomal dominance inheritance, and the average age of the patient was around 32, and the aneuploidy frequency of this patient is about 24%, which is important because these aneuploidies are reducing the number of embryo available for transfer to 50% for recessive inheritance, or 36% for dominant or X-linked diseases. Here is we performed PGT-M up to now for 315 genes. And you see that we performed this PGT in this last 9 years more than 10 times, only for 6% of the genes, in fact 18 genes, so 18 most frequent genes. And basically the remaining part are rather unique. So you can see the list of the most frequent genes on the right, and when you look a little bit more to the detail, you see that basically a PGT for 18 genes represent half of all PGT-M cycles, and the remaining parts of PGT-M for the most of the remaining genes was carried out just once. So this wouldn't be possible having the technology of karyomapping, because we would not be able to do quickly enough the setups and PGT for 50% of couples coming to PGT. So during the last years, you see that the proportion of patients coming for PGT-M without a reference sample is instantly increasing. At the moment, basically 2/3 of patients are going for PGT-M without reference samples, meaning that they know about the risk of rare disease from the carrier testing, or preconception carrier testing, or expanded carrier testing. And this number is instantly increasing. You see that in praxis, karyomapping enable us basically to provide PGT for nearly 200 new patients coming to our laboratory every year. And out of these 200 patients, 1/5 or 1/4 of them is coming with the new gene which we haven't done previously. And you see that without karyomapping, it would mean nearly 50 new setups every year, which would be hardly possible. So I would like to conclude that PGT karyomapping makes PGTM available for all patients at the risk of rare disease. Combined karyomapping and PGTA increases the chance of healthy live birth and reduce the risk of miscarriage. Karyomapping is a highly standardized and fast PGTM, a fast method. With fast PGTM preparation and communication, we prevent the risk of patient dropout and increases credibility of PGTM for patients at the risk of rare disease. Preconception carrier testing for couples at reproductive age followed by PGTM reduces the risk of conceiving child with a rare disease. And PGTM prevents the risk of pregnancy termination of affected fetus and is preferred by patients with late onset or mild phenotype diseases, typically with autosomal dominant inheritance. I would like to thank all the IVF clinics and embryology labs for sending us samples and collaborating with us. They are giving all great care to all patients and their embryos. And finally, to colleagues from my PGT laboratory. And thank you very much for your attention.